Can you help me out here? Let's see, how do we actually set this up? Oh, I see. Okay, hello everyone. I am Gatis Melkus, and today I present with some trepidation our group's preliminary results on graph-based network analysis of transcriptional regulation pattern divergence in duplicated yeast gene pairs. Or specifically, we are trying to see how graph-based analyses perform in detecting symmetry in transcriptional regulation. And this is work that we did with my colleagues in the Institute of Mathematics and Computer Science at the University of Latvia. And, well, first of all, a quick introduction. Graph-based gene regulatory networks is greatly beneficial to analyze gene regulatory networks through graphs, because they not only allow us to visualize the data, they also allow us to infer various topological patterns in this overall network data. The only question is, how exactly do we find subgraphs with some kind of functional relevance? And how do we find them within the intraspecific context? Because one thing is that network evolution has recently emerged as quite an important thing, because much of biology functions along the lines of networks. But so far there are no really coherent ways of estimating how paralogs might diverge in a given intraspecific network. And one of the basic approaches to actually analyzing subgraphs in biological networks has been through network motifs, and these are just commonly overrepresented graphlets, such as feed-forward loops, cycles. You're probably aware of many of these, and they hypothetically have a unified function, as in we can map feed-forward loops to signal delay and all kinds of other functions. But in practice, the overall interpretation of network motifs has been contentious. One interesting network motif that we're looking into in this case is the bifan motif, which is classically associated with gene duplication. Specifically, a characteristic of the bifan motif is a set of symmetrical interactions that occur with two regulated genes. And essentially, the idea is that genes with a common origin, specifically duplicated genes that once had a complete overlap of their regulatory network, should still possess some kind of overlap as time goes on. And this becomes interesting in the context of yeast, where a fairly well documented whole genome duplication occurred some 100 to 200 million years ago and left about 500 pairs of duplicates in the genome traceable to this whole genome duplication, known as onologs. And also, and the interesting part of this is that it not only left these onologs, it also left there are also quite a few tandem duplications in the yeast genome, though not quite as many as these whole genome duplicated ones. And anyway, the framework that we used to analyze these onologs was specifically the concept of symmetrical motifs, because one problem with using bifan motifs and bifan arrays is that if you take the overlap in bifan arrays, we only get essentially, we only just get the overlap of one big single input motif. However, if we embed these bifan motifs in other network motifs, the thing is that we obtain complex graphlets that we might be able to use in more complex network similarity estimations. So essentially what we do is that we embed these bifan, what we call bifan units, into these regular network motifs, and we try and figure out how prevalent they are in a given network to get a better sense of the symmetry at play. We applied this method to the yeast tract gene regulatory network, and the yeast tract network comprises about 160,000 regulatory interactions for 6,000 genes, and includes over 300 regulatory proteins that include over 70 paralogs, of which the majority are onologs. And we chose this as a point in which to check our actual method on real data, based on the previous observations that bifan motifs do indeed occur quite often in it. And essentially we used the most basic method to start with. we essentially just counted up our motifs and found that overall there are sharp differences in connectivity between different genes. Obviously, there's differences in links from each gene, and therefore, if we want to establish something about the symmetry, we need to normalize it by gene connectivity. And the idea is that this connectivity can even vary in the confines in a particular pair. For example, our symmetrical motif counts were somewhat bounded by the lower of the two motif counts for a given gene pair. And yes, essentially, once we've normalized for that, we found that overall symmetry was higher for homologs than for random pairs of similar genes in many of the motifs tested. And we observed that this homolog symmetry is overall greater for homologs too than for paralogs in general. And essentially, but this result should perhaps be interpreted with a little bit of caution because our paralog set was not especially large. But in any case, we then checked how these symmetrical motifs differ from regular motifs under simulated noise. And while the regular motifs in most cases, except in a special case of the feed-forward loop, generally stayed invariant under noise as well as in completely random data, we nevertheless observed that symmetrical motifs declined to a noticeable degree when we simulated noise. And we then tried to match it up to motif and sequence dissimilarity. We started this with the overall. We essentially started doing this with the overall gene set, which included over 500 pairs, and we got pretty excited. But the thing is that the dissimilarity of paralogs, as well as the dissimilarity of paralogous transcription factors, kind of hampered our ability to match it up to sequence dissimilarity for most of the motifs that we tested, and we observed that while 60% of sequence dissimilarity could be explained by our normalized motif counts, we still nevertheless couldn't explain a reasonable quantity of the promoter dissimilarity. And yes, essentially, there's a lot of work we still need to do on this. We need to develop more sophisticated models of connectivity and normalization. We need to figure out we need to apply these symmetry measures to other networks of duplicated genes. Obviously, we need to do a lot more work on simulated data, as well as, but also, the interesting part is that once we figured out how it really behaves in a variety of data, we can then apply it to other graphlet-based alignment methods, which are relatively scant in directed networks, as well as there being no real graphlet alignment methods for intraspecific networks. And yes, that's essentially the long and short of what we've done on this subject. And yes, essentially the takeaway here is that symmetry is a potentially relevant metric to observe in paralogous gene networks, particularly for homologs, and that we may indeed and that this may indeed be a thing to develop further. So yes, that's about the long and short of it. If you'd like to ask me some questions, I'll be here as well as at post area K27. And yeah, I'd like to thank my group and mention that our software is available on GitHub if anybody's interested in checking it out. Thank you. Well, essentially, we were wondering whether our symmetrical motifs were sensitive to small perturbations as well as we didn't want to check purely on random data. We wanted to check what would happen if we perturbed a little bit of the network. As in, we wanted to figure out what happens if say we apply this to a binding data set and we say that it's a very noisy data set. Whether these symmetrical motifs
Still find those motifs.
yeah.
If there were noise in the input data.
Exactly.
Thank you.
Let's thank the speaker.
Okay.